Good afternoon and welcome to our class in NATSI 22 or Agricultural Biology. So last time we discussed about biochemistry of cells, wherein we discussed about the different chemistry and chemicals processes of the cells, and even we talk about the different macromolecules. But now we're going to have another separate lecture about macromolecules. So from the previous discussion, we have learned that macromolecules are the big types of molecules, or the the substances which are composed of large compounds. So basically, we're studying about organic compounds as a part of the macromolecules. And these are compounds that contain carbon, which are called organic. So we have organic chemistry is the study of organic compounds. So macromolecules are large organic molecules, once again, and they have carbon with 4 electrons in the outer shell, and carbon can form covalent bonds with as many as 4 other atom elements, usually which are carbon, hydrogen, oxygen, and nitrogen. Example is the methane or the CH4. So macromolecules, once again, are large organic molecules, also called the polymers, and they are made up of smaller building blocks called monomers. Examples are the 4 major macromolecules or polymers called carbohydrates, lipids, proteins, and nucleic acids, or the DNA and RNA. So how are macromolecules formed? So this is now the topic of our lecture video on this video on how this following macromolecules are formed. So they are formed through dehydration synthesis. It's a chemical process called condensation reaction, wherein they form polymers by combining monomers and by removing water. So as shown on the figure, so one monomer to another is added with the release of water. So how are molecules separated or digested? So they are separated or digested through hydrolysis, wherein they separate monomers by adding water. So separation or hydrolyzing will cause on the on the separation of the monomers through the hydrolysis or removal of water, which is opposite to the addition of monomer, which is the release of water. So let's start with carbohydrates. What are carbohydrates and its characteristics? So basically, they compose carbon, hydrogen, and oxygen, and they contain energy with containing molecules, and some have structures, and their building block is monosaccharide, which is CH2ON with N356, and 2 monosaccharides forming a disaccharide. So these are the different forms of glucose. So these are the structural formulas of glucose, and there are 3 monosaccharides. The C6H12O, this can be glucose, galactose, and fructose. So we have learned from the previous that glucose is the sugar in blood, fructose is sugar in fruits, and so on. And the dehydration synthesis of a disaccharide, wherein glucose is added with fructose, so it becomes sucrose with the release of water. So formation of disaccharides, another example is glucose plus glucose is maltose with the release of water. And while galactose plus glucose is equal to lactose with the release of water. So hydrolysis of a disaccharide also with the with sucrose with water is where water is added, it will form glucose and fructose. The important polysaccharide starch consists of glucose subunits with plant energy storage molecule. Glycogen is a very similar molecule in animals, which are found in animals, and starch and glycogen can be digested by animals. So starch and glycogen are some of the polysaccharides that are found. Starch are found in plants while glycogen found in animals. Another important polysaccharide is the cellulose, which is composed of glucose subunits. The different bond formed than starch and its structural component in plants cannot be digested by animals. The important polysaccharides, another one is the chitin, which is composed of glucose subunits, as shown on the figure here, and partly derived from non-sugar nitrogen composed of exoskeleton of insects, and they are similar to cellulose. So carbohydrates are small group sugar molecules to large sugar molecules, like the monosaccharides or 1-glucose disaccharides or the sucrose, and the polysaccharides like the starch. So carbohydrates are, for example, monosaccharides contain one sugar unit, and the example of this are the glucose, the deoxyribose, the ribose, and the fructose and galactose. So disaccharides or two sugar unit example are the sucrose with a combination of glucose and fructose, and lactose, the glucose and the galactose, and maltose and glucose and also glucose. So they are combination of two glucose. For the carbohydrates, polysaccharide are composed of many sugar units. Examples are the starch, which are found in bread and potatoes, and the glycogen, which is found in the beef muscle, and cellulose, which is found in plants like lettuce and corn. So these are the cellulose glucose, which are composed of many sugar or glucose units. So let's move on to the lipids. What are lipids? They are composed of carbohydrate, hydrogen, and carbon, hydrogen, and oxygen, which are greater than 2:1 ratio of hydrogen is to oxygen. This includes the fats, the oils, the phospholipids, and cholesterol. And these are the building blocks, fatty acids, and glycerol. Energy, they are source of energy and storage molecules, and phospholipids even are part of the cell membrane. So this is the fatty acid structure. As you can see, the carboxyl group, COOH, it forms the acid, while the R group is a hydrocarbon chain. So this is an example of representative of fatty acid. This is the saturated fatty acid, like the palmitic acid, and the unsaturated fatty acid, or the linoleic acid. So as you can see, the difference between the two is very clear in terms of structure. As you can see here, the double bonds, and here the. So for this is the structure of a glycerol, and fats are synthesized as shown below. So glycerol plus fatty acid is equal to fat with the release of water. So this is an example structure of a phospholipid, which are found on the membranes. And these are the lipids, general term for the compounds which are not soluble in water and are soluble in hydrophobic solvents. Um, they store the most energy, and this is examples are the fats, phospholipids, oils, waxes, the steroid hormones, and the triglycerides. So let's talk about the lipids functions. So there are 6. They are for the long-term energy storage, for the protection against heat loss or insulation, for the protection against physical shock, protection against water loss, and chemical messengers for hormones, and also the major component of membranes known as the phospholipids. So example of lipids triglycerides are composed of 1 glycerol and 3 fatty acids, as shown on the structure figure here. So fatty acids are of 2 kinds, the saturated fatty acids and the unsaturated fatty acids. So for saturated fatty acid, don't have double bonds, and they are marked as bad, but because of they are so saturated, while unsaturated fatty acids have double bonds and they are marked as good fatty acids. So next are proteins and their characteristic. Again, they contain carbon, the hydrogen, oxygen, nitrogen, and sulfur, and serve as structural components of animals. They serve as control molecules for enzymes and transport and messenger molecules. Also, building block of is amino acid. So this is the structure of the amino acid, wherein the amine group, like a base, tends to be positive. The carboxyl group acts like an acid and tends to be negative. And the R group is a variable from 1 atom to 20. So there are 2 amino acids joined together to form a dipeptide. And the adjacent carboxyl and amino groups will also band together. So these are examples of amino acids. We have the glycine, the alanine, the valine, the leucine, isoleucine, the methionine, the phenylalanine, tryptophan, and the proline. Some more amino acids are also serine, threonine, cysteine, tyrosine, asparagine, and glutamine. So there's still more amino acids, and there are even 20 more amino acids like the aspartic acid, the glutamic acid, the arginine, histidine, and lysine, which are very important for body. That's why if you are vegetarian, one thing that is lacking in your diet are some of the amino acids which are not found in plants, because some of the amino acids are found on animal diet. So how does the formation of the dipeptide? So this is through the dehydration synthesis. So again, with a combination of 1 amino acid to 2 amino acid, and with the release of water. So dipeptide is 2 amino acid, while tripeptide is 1 amino acid and 1 dipeptide, while polypeptide contains many amino acid and tripeptides. So a protein consists of 1 or more polypeptide chains, looks like this. So proteins or polypeptides contain amino acid, which about 20 different kinds bonded together by peptide bonds. So it has 6 functions: for storage, like the albumin, the egg white, and the for transport, like the hemoglobin, and for regulatory, for hormones, and movement, for muscles, and for structural for membranes, hair, and nails, and for enzyme, cellular reaction. So these are the 6 major functions of proteins. Again, storage, transport, regulatory movement, and structural and for enzyme. So there are 4 levels of protein structure. We have primary, secondary, tertiary, and quaternary. So the primary structure, where amino acids banded together by peptide bonds straight, it only consists of a straight chains. While the secondary structure is a 3-dimensional folding arrangement of a primary structure into coils and pleats held together by hydrogen bonds. So with an alpha helix or beta pleated sheet. While the tertiary structure or secondary structure bent and folded into a more complex 3D arrangement of linked polypeptides. We have the hydrogen bonds, the ionic and the disulfide bridges, and these are called the subunit. Alpha helix and the beta pleated sheet are combined together. And the quaternary structure are composed of 2 or more subunits, which are globular in shape and form in the aqueous environment, like the enzymes hemoglobin. So these are the subunits. Next are the nucleic acids. So these are the 4 types of macromolecules, the chemical link between the generations, the source of genetic information in chromosomes. So they dictate amino acid sequence in proteins and give information to chromosomes, which is then passed from parent to offspring. So what are they made of? They are made up of simple units called nucleotides connected in long chains, and it has 3 parts, the 5-carbon sugar or pentose, and the nitrogen-containing base made of the carbon, hydrogen, and nitrogen. It has a phosphate group P groups, which make the links that unite the sugars, hence they are sugar phosphate backbone. So there are 2 types of nucleotides depending on the sugar they contain. We have the RNA or the ribonucleic acids. It's the pentose sugar is ribose and has a hydroxy group in the 3rd carbon OH. While deoxyribonucleic acids or DNA, the pentose sugar is deoxyribose and has hydrogen in the same place, deoxy minus oxygen. So there are 3 parts composition of DNA nucleotides. We have the deoxyribose sugar or no oxygen in the 3rd carbon and the phosphate group and one of the 4 types of bases, all containing nitrogen, adenine, thymine, cytosine, and guanine. The thymine only in DNA. The RNA nucleotides also compose 3 parts, the ribose sugar with oxygen in the 3rd carbon, and phosphate group, and one of the 4 types of bases all containing nitrogen, adenine or uracil, cytosine, and guanine. Uracil is also only in the RNA. So what's the difference between the DNA and the RNA? DNA has, of course, because of their pentose sugar, they have deoxyribose sugar, while RNA has a ribose sugar and bases adenine, thymine, cytosine, and guanine, while the basis is also the same except for uracil. And DNA is a double-stranded helix arrangement, while RNA is single-stranded. So the double helix DNA structural model is proposed by Watson and Crick in 1953. The 2 sugar phosphate strands next to each other but running in opposite directions. So they have specific hydrogen bands occur among bases from one chain to the other. So we have from A to T and C to G, and this is due to the specificity of a certain base on one strand, which indicates a certain base in the other. So the 2 strands intertwine, forming a double helix that winds around a central axis. So how does DNA work? So DNA stores genetic information in segments called genes. While the DNA code is in triplet codons, short sequence of 3 nucleotides each, if certain codons are translated by the cell into certain amino acids. Thus, the sequence of nucleotides in in the DNA indicate a sequence of amino acids in a protein. So nucleic acids are of two types, the deoxyribonucleic acid or DNA double helix and the ribonucleic acid RNA single strand. And the nucleic acids are composed of long-chain nucleotides linked to dehydration synthesis. So nucleic acids or nucleotides include phosphate group, pentose sugar, or with 5 carbon, and nitrogenous bases adenine, thymine, uracil, cytosine, and guanine. So this is the structure of the nucleotide, the phosphate group, that's sugar deoxyribose, and the nitrogenous base AGCT. And this is the double helix DNA. So I guess that's all for this video on the different macromolecules. So if you have any questions regarding this video, please just comment on the posted video, and I will see you again on the next video for another discussion. Thank you very much for watching this video.